Hey, I'm Mike Solana. Thanks for checking out our live stream at Founders Fund on synthetic biology and longevity, which we're going to be talking about today. I'd like to just quickly introduce my panel of ingenious friends and colleagues here.
Hi, my name is Anselm Levskaya. I've been working in engineering mammalian cells and synthetic biology for maybe 15 years now.
Hi, I'm Laura Deming. I run a venture capital fund focused on investing in companies that aim to extend the human life.
And I'm VanDevender. I'm chief scientist here at the Founders Fund.
All right, before we get started, I want to kind of just quickly frame the conversation. I think a lot of people don't believe that extending the human lifespan, longevity, is a serious question. Or you talk about it, they kind of don't understand what you're saying. And that's very much what we're going to be talking about over the next 45 minutes. It's just that this is a problem that can be solved. And we're going to kind of come at it from a bunch of different directions. I want to start with just maybe talking about what the big goals in synthetic biology are, kind of give everybody a kind of that question. Anselm, like, what is the holy grail here? What are we actually working towards? Why is this such an exciting field?
Well, in biology, it's really it's the biggest reverse engineering project in the history of mankind. We're trying to reverse engineer this 4 billion-year-old alien technology, and we've come a long way in the last century. And in synthetic biology, the real goal is to be able to make predictive changes, to actually be able to engineer these cells that build and make up our body, and to do so in a way that allows us to treat disease and perhaps one day soon even enhance natural human capabilities. But along the way, what we're figuring out now how to do is how to actually specifically in a safe way engineer cells to achieve some medical goal right now, and to use them as sort of a new form of therapeutics. It's sort of my view of the situation.
Laura, is this pretty much how you think about it as well? Or?
Yeah. No, I mean, I think synthetic biology, like, one of the coolest things about it is, back at MIT, I worked in a lab for a bit, and we literally were like, 3 kids for a summer, like, what if we just trained cells to, like, respond to pressure and, like, create bone? Like, could we make, like, an armored thing that somebody could wear, and, like, they get thrown in a car, and then, like, they get a bone protective suit, and, like, obviously we didn't get quite that far along, but it's just amazing how quickly, as, like, a kid, you can just take stuff and in a summer, like, make something that's actually insanely cool, like, in biology. And I think that's really recent, and not quite in the popular sphere as much as it should be like right now.
It seems like people throughout time have thought of biology as this fixed thing that maybe you could prod and poke and adjust slightly, but we're now talking about reconceiving what it means to be.
Yeah, we literally have, like, parts. Like, we have a parts library, and we can, like, put the parts together to, like, make actuators and stuff, and it's, like, really cool existing technology.
So what do you see, like, what is endgame? What is the ultimate goal? What does perfect biological control look like?
Lego blocks, like, you know what you want, and it's very straightforward. And I mean, like, then it's very complicated, of course, because these systems are very complex. But I think the thing that I love about synthetic biology is just the engineerability of it. Like, that's the cool thing, for me at least.
Aaron?
Yeah, synthetic biology is great in that it is a better understanding of what it really means to be a living organism and what it means to have a disease or old and die. We've had medicine for a long time, and the medicines that we give are just simple molecules, and those are static things. And if your understanding of a disease as this is just a static condition, like you have a bacteria, right? That's a fixed state, and I give you some antibiotic, and then I can change that. But if your understanding of disease is more sophisticated than just like presence or absence of a pathogen, and that it's really about how all of the systems in your body is working together, then being able to treat that with just a molecule that's a static fixed thing is really too limited. So synthetic biology allows us to create living dynamical systems, i.e. a cell, which is a self-contained living dynamical system, that we can program in just in the same way that a synthetic chemist can make molecules, a synthetic biologist can make cells or biological systems that can treat the disease, which is a part of your living condition in motion. It's no longer just a static model for what it means to be well or sick. It's now a dynamic model of your whole metabolism functioning together and using these dynamic tools like cells, we can synthesize ways of addressing those in motion.
What would you say are some of the huge problems that we're looking to tackle with this kind of stuff?
Probably the best, most straightforward one would be like diabetes, right? Which is a metabolic kind of issue where you have your blood sugar and your insulin are always chasing after each other. And so we've been trying to treat that for years, since for 100 years by the static application of insulin, but it never really works out. It's very difficult and it's a very burdensome disease to manage where you're testing yourself multiple times a day, injecting yourself multiple times a day, because fundamentally you're trying to alter a dynamic disease state, you're changing insulin level with a static test and drug.
Anselm, this sounds somewhat similar to what you're working on. You guys are targeting diseases and reprogramming the human immune system to go after them. Can we talk a little bit about that and just kind of what are the interim steps to kind of perfect biological control?
Yeah. Well, right now people are really interested in the immune system mainly because it's a very easy system where you can take the cells out of the body, treat them, passage them, make genetic modifications to them, check them out, and then put them back in. And the immune system is naturally diffuse and it works sort of as a distributed system. So it's a very good place to initially begin trying to do real genetic therapies for human diseases. Diabetes is really interesting because it's a very simple disease in some sense that type 1 diabetes comes from.
Let me just make sure I understand this correctly. So you have a disease, let's say, give me an example, like...
right now we're focusing on cancers.
So you have a certain kind of cancer, then someone from your team or someone will come in and actually change the way that your body reacts to sort of foreign bodies and kind of go after the cancer.
What you're trying to do is really sort of just teach the immune system that the cancer really is a foreign entity that needs to be destroyed. And so what people are doing right now, they're trying to teach T cells, they're trying to engineer the T cells to recognize the foreign presence, it's not the foreign, but it's the rebellious presence inside you, to detect the cancer cells and to just annihilate them. And this happens rarely, naturally, but it's typically not effective enough and for your natural immune system to clear your body of the cancer. But what people are trying now is to sort of accelerate the process of just teaching your immune system to recognize these foreign cells and to wipe them out.
What are some of the approaches to this? How do you get your immune system to do what you want it to do?
Well, what you do, you sort of, there's an incredibly complicated and beautiful natural system for discovering foreign antigens, like foreign shapes on cells that are not your own, or bacterial cells or viruses. But your immune system really did evolve to get very good at detecting viruses and bacteria. It's not so good at noticing when your own cells have gone rogue. In fact, it sort of avoids attacking them because that's the basis for autoimmunity. So what people are doing right now, they're basically short-circuiting the whole natural process of discovering what's foreign. They literally they design a receptor, to design a receptor with a protein that is designed to bind, to literally stick to the cancer cell. And so what you're sort of very quickly just teaching the T cell, this is what you want to go after. You want to go after this guy, and when you see it, you kill it. And so we're sort of short-circuiting the natural process of antigen discovery and presentation, just sort of directing them by hand.
I'm wondering, Laura, you're a biologist and you're also now a partner and the founder of the Longevity Fund, so you're investing in this kind of stuff. How are companies dealing with these problems, thinking about these problems, and how do you think about investing in companies like that?
So one really cool thing about this field, right, is synthetic biology is not like a recent phenomenon. Like we think it is, we're talking about it like it happened in the past 5 years, but think about the 1980s, right, like the founding of biotechnology as a whole, that was recombinant DNA, like that was copy-pasting some DNA into some cell and having that cell create the protein that you wanted to put into a person. And we're talking about insulin, like how insulin's not a great cure for diabetes, and it's not, but it's incredible to me that for 20-something years, we've been taking human cells and getting them to make this protein, which is extremely complex, purifying that protein out and then taking it into a human, and that's really kind of synthetic biology, right? And so when Anselm's talking about these cells that are very good at binding to kind of these foreign entities, that's antibody technology, and that's the founding of biotechnology, in the 1980s again. That's Genentech saying, what if we just took some piece of a cancer and just put it into a mouse and had that mouse figure out what the heck was going on, try to attack that cancer and kill it. And then sort of develop these really good binders for the cancer kind of molecule. And so, when we talk about synthetic biology, yes, it's like the field of complex synthetic biology, of taking multiple parts and putting them all together. That's very recent, but kind of like the field of manipulating cells in this way is actually kind of, I think, it's been the birthright of biotechnology since it first started about 30 years ago.
I would say there has been another, it's more than just the complexity. There's a phase shift in that the making recombinant insulin or making antibodies from CHO cells is definitely synthetic biology, but it's sort of like ancillary synthetic biology because it only happens in a dish. And then we maybe take the results of that and we put it into your body, but the way that when it actually, like, the rubber hits the road, when the needle hits your vein, that is still static molecules. The region we're getting into now is where we're taking those reprogrammed cells and applying them into your body directly. So it's like primary synthetic biology medicine rather than secondary.
What would you say are, I mean, we've had, around here in Silicon Valley, we've had this tremendous wave of incredible companies emerging from computer sciences, a lot of software companies and hardware companies. How does biology fit into that? What are the differences maybe between starting up companies in both? Are we poised to see another wave of maybe now biology companies as it becomes a lot cheaper to do this stuff? I mean, what does that kind of stuff look like?
I would say we are poised for that, and there are a lot of reasons for that, but the one I want to highlight right now is the predictability of biology has come a long way in the last few years, and that one of the reasons why Silicon Valley was really able to create a lot of these technology companies very quickly was that you could do things cheap and you could do things reliably, because when you run a program on a CPU, on a computer processor, it runs the same way every time. And the tools that we had for doing biology were very noisy. And so that prevented, whereas when you're, say, studying physics as the science and using that as a foundation for engineering, or computer architecture as the foundation for software engineering, biology wasn't at the point where it was reliable enough and cheap enough to become a really good foundation for biological engineering. And that has really started to change. And that we've seen a lot of lab-scale automation kinds of things happening, and also just our basic fundamental understanding of things like genetics from cheap sequencing, and the tools that we use to understand the biology have sort of gotten us to a place where it now makes sense to really think about biology as a mature enough science to use as an edifice for engineering.
Cool. I kind of, one thing I think we have to talk about in this conversation is just CRISPR-Cas9 and gene editing in general. Can you just give us a quick, Anselm, do you mind giving us just like a quick, for our viewers, like what is CRISPR, why is it important, how does it fit into the sort of other gene editing techniques?
Yeah, so one of the biggest challenges, say you know that there's a mutation in a person's cell that is causing a disease, cystic fibrosis is caused by one mutation. You'd like to go in there and just fix it. You'd like to go in there and change it. Or say you want to put some new stuff in, but you don't want to put it in the middle of a gene and destroy that gene. You want to put it in a very specific place. That has been an incredibly hard thing to do. Just the ability of targeting an edit. So being able to go into one small place in the 3 billion letters that makes up your code and make a change right there. What people have done for most of the last few decades has been throwing stuff in kind of randomly. And there's a few exceptions in mice, for whatever reason. Actually, there's a trick that lets you sort of edit them somewhat decently, and that's why mice have become such a huge research model, because this is so much easier in them. But in general, it's incredibly hard to do this in higher animals, in mammalian systems. And so what CRISPR lets you do is to basically very specifically target a little protein that just cuts the DNA in a particular place, and that recruits all of the repair machinery that your cells use, that will then integrate in a little piece of DNA that you also throw in the cell, and just it'll basically repair in, clip in your edit, and create that alteration. And so being able to target, being able to actually say, I want to change that right there, is a huge deal, and it really is enabling a lot of new research.
So if right now, today, we are able to just target a gene and cut it out or change it, what problems are left to solve, right? That seems pretty great, right? Like you should be able to just control the genome theoretically. What is preventing us from doing that?
There's two things. Even as good as CRISPR is, it still needs to be improved a little bit. It still will occasionally cut places you don't want it to. It's so new. There's still a lot of work happening. That progress will be made very rapidly, where we'll get it to be much more specific. We'll use some other tricks, and we'll make it really clean so that you can feel quite confident you're only targeting the change you want to. But aside from that, the real challenge is just knowing what to do. I mean, some problems are very easy. Cystic fibrosis is very simple. You know what the problem is. You know it's a single point mutant. You know that you just want to revert that to the natural wild type allele. But the challenge there is that you're typically dealing with that mutation in a bunch of epithelial cells in your mucosal lining, and you want to find a way to get this CRISPR and the altered gene into those cells. So there's still a huge problem with delivery. So traditionally we've been using viruses to do that, and they work pretty well, but they're not quite good enough yet. And it's really sort of packaging CRISPR along with all of these viral delivery technologies to make these edits. And that's just talking about making, just fixing known errors. When you want to talk about doing stuff that's more synthetic, where you want to bring new stuff in, the challenge there is really figuring out what are you going to put in? Like, what is the smart thing to put in? There's still a huge amount of work to be done to figure out, what am I changing? What actual new stuff do I want to put in to address a particular problem or do something new?
You said 3 billion.
3 billion letters.
That's the letter. Each one of us has a sort of unique 3 billion letter code.
There's a few megabytes of difference between us. Yeah.
We don't. What is roughly the fraction of those letters that we actually even know what they do? I mean, how many of those things do we understand?
So if you're talking about the protein coding genes, which is a small fraction, we know what a lot of those proteins do. There's still a lot we don't know, but the real sort of dark matter in biology is understanding what's called the transcriptional regulatory logic. So this is, the gene is the what, but the where and the when. Like, when do I make this gene? When do I make this protein? Where? At what time of my life? When I'm a child or an adult or as a developing embryo? There's an incredibly complicated state machine, a very complicated dynamical system that is encoded in these promoter regions, these non-transcribed regions. They're regions of your DNA that don't encode proteins, but they encode a kind of logic, a kind of developmental code. So a lot of what your body plan is encoded in this, regulatory logic, this just promoter stuff. And we are getting better at sort of beginning to read what some of that stuff does, but I'd say our understanding of that transcriptional logic is still fairly primitive. You can't go in and just look at the code and say, oh, that's going to cause me to have a second thought. It's going to change the Hox gene's expression pattern, and it's going to cause my limbs to look a little different. And so much of the code that dictates how complicated bodies work is really stuck inside that non-protein logic.
What is it going to take for us to understand that, to know, I mean, what are the steps here to understanding the genome?
Yeah, I mean, there's a lot of work going on in this direction right now, and particularly when you're talking about this transcriptional logic in the context of, say, development, when just to be concrete, and when you're talking about how do bodies develop from a single cell into a complicated machine, a lot of the work that's happening right now is benefiting from the fact that we have amazing microscopes nowadays compared to what we did even 10 years ago. And you can actually look in like a zebrafish or a small developing mouse embryo, you can actually watch all of the cells in the body up to a certain point. Every single one of them, you can begin watching how they divide and grow, and you can also use all of these really impressive fluorescent proteins that you can in these research models, you can splice these proteins in, and you can watch how particular parts of this complicated transcriptional network, how they come on and off, blink on and off over time. So you can actually begin just watching it work. You can begin taking measurements of this dynamical system in action. And that, in a sort of high throughput way, will really allow us to begin getting the data sets we need to sort of reverse engineer what's going on with that logic.
We talked earlier, all of us talked a little bit earlier about needing data. We need access to large data sets. We need to be able to see what everyone's genome is, so we can compare it and learn what these things are. in this country right now, maybe Laura, do you mind walking us through
Yeah,
the
sure.
process of,
Love to.
I mean, if we were trying to figure out what the genome is, we need data to do that. Can you explain maybe why, and then what that process looks like? Like, how do you access that data in this country today?
So if you think about like genomics in general, it's kind of like the most perfect machine learning problem you could come up with. You know, it's like literally a one hot encoded vector like given to you, like you have 4 possibilities of precision. And like that's just crazy, right? It's like nature gave you a machine learning problem. It's like, here, do this. But the problem is like, you know, because we only have 3 billion base pairs of information per person is a lot, and to really get statistical kind of power out of that, you need on the order of maybe tens of thousands at minimum, hundreds of thousands kind of at maximum sort of genomes to work with. And we really, today in the world, I would say there are probably about 1 million genomes fully sequenced that exist, spread across many different countries and agencies. In the UK, there are about 70,000 that are kind of publicly available to researchers there. It's harder to get that data here. The US has about 400,000 veterans that they've taken blood samples from, but only about 2,000 of those have whole genome sequenced. And the US also has approximately 10,000 to 40,000 kind of whole genomes that are kind of locked up in this database called dbGaP, where if you apply for it and you're a professor, you can kind of get access to it. But I think a really big problem is, this is really a machine learning problem, and you can actually throw kind of basic image kind of recognition nets at genomes and immediately get like state-of-the-art results in biology, which is pretty crazy. And you can do this with any kind of net that you can think of, recurrent, et cetera, for most kinds of problems you can think of in biology, which is a really amazing fact. But all the researchers that you talk to in this field of machine learning who are passionate about biology, maybe have kids with diseases, they're like, but the data is just not there. Like, I know it's there, but like to access it, I would have to write forms and talk to administrators, and I don't want to do any of that. Like, leave me to do my models, and you go get the data, and nobody wants to do that. So it's pretty crazy because, in places like China, I think, for example, you're seeing this amazing openness to just give data to everybody, like, send it to the US, send it to whoever has the technology to analyze it. And in the US, it's kind of like, no, we have to be very, very cautious and careful, and that's great, but it also is kind of, I think, putting us at a disadvantage to a lot of other places.
Why is that?
Moving more quickly.
Why in this country are people so reluctant to, reveal their genetic code?
Well, I mean, would you put your genome online today? And like,
So I would, until you mentioned the insurance thing, which I hadn't even thought of.
I mean, technically, I believe it's true that there's actually a law that was passed maybe a few years ago that forbids insurers from using genetic data for making decisions. But it is generally a worry. Your genome.
The fear is that like your insurance company sees that you have genetic predisposition to, some crazy
Huntington's disease.
Huntington's, for example, and they just don't want to give you insurance.
Yeah,
so now there's a
rather
law.
not.
There's a law against that, but
There's a law against
the fear is maybe that's.
Perhaps they'll find a way around it. Perhaps it's not going to be enforced. How can you prove that they're using that information? There's genuine concern in America because of the way that we insure ourselves and get health care and pay for health care. That makes people very leery about just putting up their genome online. And the genome is really only part of it. I mean, it's an important part of it, but if you just have a string, literally just the code, it's only moderately useful. What you really want is to have both the genome of the patient as well as a lot of basic lab tests, and hopefully longitudinally over time. You'd like to know some of the patient history. You'd like to have a whole family's genomes and basic phenotypes. So we say in biology you have the genotype, which is literally just the string. What is your code? And then the phenotype is like what's the color of your skin, the color of your hair, your eyes. Do you have these diseases? What's the sum total of your actual, how does your body work, And what you really want when you're going to apply machine learning models is to have not just the genome, but also a lot of facts about the patient, as many as we can, basic blood protein levels and other, everything that a doctor might have in a patient record. You ideally have some sort of anonymized form of that to be able to begin doing supervised learning on.
Laura, you were just saying that, if you had all that information, you could run it through a computer now and see some pretty interesting results.
It's crazy what you can do with like the crappiest information possible. Like today people are working with on the order of like thousands of publicly available genomes, and like they're able to predict like basically everything that you can get from these publicly available data sets, like with very high precision with nets. And so it's not hard to think of like all of these you could start to approach better.
So we need
with that. Yeah.
So we need more data. What are some workarounds, Aaron?
We need access to the data.
We need access. So we have lots of data. We need access to the data.
Yeah.
Well, there's two barriers to it, right? And one reflects the other. So privacy is a very strong cultural value, more so in the West than in places like China. And so we need policy systems that reflect that cultural value just to be able to past snuff or existing in the wild. One of the ways in which that value is coded right now is that in order to mess around with people's data, you need to be able to argue that there's some tangible benefit to that patient, right? So when things are purely exploratory, there's a lot of resistance to that because it's hard to articulate. I'm putting some people's privacy at risk. They're taking some exposure, but I can't articulate a specific benefit until I actually, run my nets and figure out what the upside is, right? So we need a way of encoding that benefit to people that in a more abstract way, so that we can create a sort of policy framework that says there is huge advantage to everyone, including the people that are part of these databases, and that allows the researchers to go in and start looking at things in a way that justifies the privacy exposure. Once you do that, then it makes it possible to develop the technical systems to actually make the communication possible, right? Like now, all these different countries and agencies, they've been growing organically, and so they've put all of this database, all this data in just homespun systems that are protected because there's no policy impetus to do that large scale kinds of sharing. But if you have that, then that creates an environment where it makes sense to actually do the technical work to put it all together.
Why is there no push for this from either? There's no cultural push, there's no government push. Why are people not thinking about this in this sort of serious way?
I think people are thinking about it in a serious way for sure, and it's mostly local level decision making, right? Especially in this country where the people who have the data have to build coalitions in order to do that sorting. In the UK, they have a National Health Service, and so there is a top-down organization that can store everyone's genetic information and make sure that it's used appropriately. In the US, though, there's always the concern that, okay, if we choose anyone to sort of be the one true place that everyone has stored it, that person or that organization might derive some preferential benefit, and then the people who gave them their data lose out. So figuring out a way to solve that coordination problem so that everyone's interests are aligned is very difficult and very political. People are absolutely working on it, but it's not something that happens quickly.
Especially not in this country, it seems. You're saying, Laura, that you were afraid that we were falling behind, for example, like China because of this.
I don't know, I think China hopefully will collaborate with us to do things, right. But it's just weird to see such an advocate, like everyone wants to do this thing, but then everybody just can't quite agree, and so then we don't do it for 5 years, and then we're quite behind on a lot of different measures. I think another thing is that, when we sequenced the genome in the early 2001 or so, I think a lot of folks, there were covers on Nature and Science, and people saw that the genome had been sequenced, right? And so obviously if genomics was a real thing, we'd have many diseases cured, and we didn't immediately after that fact. And so I think a lot of folks, if you took somebody on the street and you said, we have this new CRISPR therapeutic and it's great, they'd be like, yeah, I've read about that recently, maybe. But if you said genomics, they'd be like, well, didn't we do that, why now, why me, et cetera. And so it's hard to get people, it's amazing to be we got 400,000 veterans to just be like, yeah, sure, take my information. I've done a lot for my country, I'm going to do some more. And then other folks, I think it's a different, have a different approach to it.
It's interesting that you mentioned maybe sort of, so we cracked the human genome in the early 90s. It was 90, what was the year?
I think 2001
we started in the early 90s. It was launched in the early 90s, it was cracked in the early 2000s. And there was this wave of press that said, we would have cures to sort of every disease.
There were covers with people's faces on them, we're going to understand aging, what makes you...
Why a pattern? Maybe, just what is...
Because you have 3 billion base pairs. What do you do with, you just given A, T, C, G for 3 billion positions. There's nothing you can do with that at all. You could look for patterns, but even without machine learning at that point in time, it would have been really hard to just unstructured, in an unstructured fashion, find patterns. And so now we can kind of do that, but to do great, work types.
If I gave you the binary compiled version of Linux, say modern Linux, and I just gave that to you and said, tell me how it works. works.
Well, I would give it to Aaron.
Even that would take you quite a few years to really understand all the intrinsic structure and order inside that beast. And unfortunately, with human genome, we don't have, the source code wasn't included, fortunately. So, it's even harder because the system that's running it is not as deterministic. It really is, the way that biological cells work, and proteins work, there's a bit of a statistical nature to it, and a very subtle one. And ultimately, a lot of the binding interactions between proteins are quantum mechanical in nature, and they're very, very hard to predict through straight brute force simulation. So it's really very hard to just pull out the implicit logic that's occurring inside the dynamical systems inside cells. Even if you isolate yourself to just talking about a small given system, a few proteins interacting together, it can be quite hard to predict things from just the code. And so the real challenge, is that the code is not enough. It's necessary, and it was a critical thing to do, but we've got to do quite a lot of experiments on top of having that code to really begin figuring out how this stuff works.
I would say there's even a deeper problem to this, which is that the metaphor of thinking about it as code running on a computer is slightly wrong, because if I look at the Linux source code, I can be said to have understood it if I can tell you what each piece is for, i.e. what the intent of the programmer who wrote it was when they wrote that piece. But there's no writer from our genetic code. There is no intent. It's an evolved system. And so we can impose our own interpretations of what things are for, but that doesn't actually map to the underlying intent from an original programmer. And so thinking about it that it has to have that level of abstraction just doesn't work. There's parts in there that are, that are essentially chaotic from our level of interpretation, right? And so we need a way of making predictions about what will happen without getting drawn into this. This is how the code is for. This is an understanding human consciousness level version of what is actually happening.
Cool. Well, we got a couple minutes left. We've kind of outlined the big, huge goals: controlling human biology, ending aging, hopefully one day, in the short term, ending getting rid of diseases. We have this problem of data. We have a lot of it. We don't know how to necessarily figure out what everything does. If we don't have access to the different pieces, there's this difficulty in understanding how to read it. If it's, again, if we're applying our own language to this system that really is not, maybe it's not, perfectly analogous to computer programming or something. What are the signs for hope? There's like, it seems like a complicated situation. What do we have to be excited about?
Yeah, so the big thing that's happened in the last few decades has really been better computers, and really amazing imaging and sensing technologies. So again, the fact that we have computers that are fast enough to really crunch through the massive amounts of data that we're collecting, not just from genomics, but also from brain imaging and from just watching cells in action using fluorescent recorders, or just gathering these very large, image and video data sets that sort of are a lens into what's happening inside cells. The fact that we have these sensors that can do this, that are sensitive to a single photon, and we can just stream this information in terabytes a minute into GPUs and actually crunch data, reduce it, and begin looking for real patterns in it. I think the fact that we have such amazing tools for measurement, and they keep coming out. We have sequencers that are incredibly cheap to run right now. In fact, with nanopores, you can buy I think what is it, $1,000 sequencer right now, and actually run it yourself. It's very impressive. And these things are only going to get better and better. And the same nanopore technology might soon allow us to actually directly sequence proteins and to do surveys of what's inside a cell in a very fast way. And these things they're going to keep coming out. So a lot of the hard tech is invading biology and really giving us many new viewpoints and lenses. And it's making data much more cheap and much more reliable and regular and repeatable than what we've ever had before in biology. And so just our sight into the system is growing very fast. It's the thing that gives me a great deal of hope in actually being able to understand what's going on inside cells and to be able to predictably change them. Laura?
Cool. Yeah, so I think just from the aging aspect of it, because this is my true love and where I come from, like, in the 2006 to 2009 era, kind of when you were doing aging in a lab, you were really taking worms and you were like, if we change one gene, like, what happens to this worm? Like, if we change two genes, oh, like, that's a big deal. Like, put two things together, see how long it lives, and we're getting, like, 10x or so increases in lifespan, doing that kind of thing. I think it's really interesting to kind of see how that's evolved to the clinic and kind of see a lot of companies coming along. And our fund invests in some of these, but, like, a lot of companies are targeting the same pathways that we were working on kind of in that family, or kind of that we see in different models of aging. And maybe most of them will fail. Like, there are about 50 different ones that I would say are reasonable guesses, but kind of like you have this feeling of hope that, like, maybe the first therapeutic that actually affects lifespan, not like 2x, maybe more like 30%, 50% max if you like combine a bunch of things together. Maybe those first therapies that affect lifespan and are affecting aging are kind of already on their way and in the clinic. And so I think kind of what we're talking about today is more, like, if you think beyond just kind of the single gene kind of focus on the kind of limited view of biology to date, what more gains could we get if we looked at things from a systematic view? And that's a really exciting and totally unknowable kind of open field, right? And that's kind of where we're heading. So it's an exciting time to be in biology in general, I think.
I am very optimistic about it in that we've had medicine for thousands of years, but medicine has been only a top-down empirical pursuit thus far. You try a bunch of things, you see who gets better and who dies, and you go with what works, right? When Hippocrates was building a hospital, they hung some stakes in different parts of the town, and the one that developed maggots the slowest is where they built the hospital. So they didn't understand what maggots were or why that worked, but it's just sort of that's what works, and so they went with it. And we developed things like the double-blind study and doing trials and made it more rigorous, but fundamentally was still a top-down pursuit. And now we're getting to the point where we can do biology and medicine and health on a bottom-up sort of way, where biology is getting to the point where we can really start to understand things from first principles and then not just deal with what works, but construct the physiology and the anatomy that we actually want to have.
Thanks. Thank you guys all for joining us, and thanks for tuning in.
Yep.